I trained under 2 co-advisors. One, his name was Dr. Merritt Kimmel. He is like an expert in branching processes, statistical genetics, molecular evolution, bioinformatics. And then I also had a second co-advisor at Texas Children's Hospital and Baylor College of Medicine, Sharon Kwan. If you are familiar with Houston, Rice University is on one side of the street called Main Street, and then right across the street is what's called the Texas Medical Center. It is one of the world's largest medical institutes with over 60 medicine-related institutes, universities, and hospitals, with the largest cancer center, MD Anderson, and one of the largest children's hospital, Texas Children's Hospital. So during my time there, I actually went and pipetted in my co-advisor's lab, and I learned the value of how hard it is to generate really good biological data. I also modeled genomic data from pediatric tumors, so I really started to fall in love with biology during my PhD. And I thought this would be just like a fun fact about me. This is not my first time to the University of Washington. So back in 2011, I visited as a graduate student, and I completed one of the summer institutes in statistical genetics course. You can see Bruce's signature down there at the bottom. And just in case you're interested, this is me when I looked like a child with really short hair. This is my husband came with me, and we had a really great time. Okay, so after that, I went on to do a postdoc in Boston at Dana-Farber Cancer Institute and Harvard School of Public Health with Raphael Irizarry. And while I was there, in addition to the genomics, which I'll talk about in just a second, I had the opportunity to teach data science with him, both at the undergrad level and at the School of Public Health. And he is passionate about a sport called baseball. So he really likes to teach statistical concepts through the sport of baseball. So while I was there, I attempted to learn about the sport. It was never something that I was really into, so I attended some games. This is me attempting to learn about baseball so I could better understand whenever we talked about hitting averages in the context of empirical Bayes, what is a hitting average? So lots of fun. So in addition to the teaching, during this time, there was a really big revolution in the kind of data that we could generate. So across these 6 human tissues, for example, I hope you can appreciate the cellular diversity between the epithelium, the intestine, the liver, the blood vessels. Cell types are very, very different. They all have different cell states, cell interactions, and the technique that was invented around this time in 2014, 2015 is what we know today as single-cell RNA sequencing. And the idea was simply we could take each individual cell and profile the molecular genetics or genomics that were being expressed in the cell at the time. So you could essentially take the cell out of the tissue and then sequence all the RNA in that cell. So this was really, really exciting. We started out with just being able to sequence a couple hundred cells, and then it was quickly a couple thousand cells. We can do things like the following, where we can take a heterogeneous group of tissues and we can apply dimensionality reduction techniques and project high dimensional data, meaning each cell there has up to, say, for example, 20,000 protein coding genes or features that we can profile. We can project all the cells, every dot there is a cell in a low dimensional space, such as along principal component 1 or 2, and we can start to see how the cells will separate. We can apply supervised or unsupervised learning algorithms to annotate the cells. For example, are they in a particular cell state? Or are they a particular cell type? We can project cells along different kinds of lineages. Maybe they're going through some kind of differentiation process, and we're interested in understanding the different stages of the differentiation. Or maybe we're interested in understanding the spatial resolution of the cells. Lots of cool things we could do with the data. So formally, single-cell RNA-seq is the following. You take a piece of tissue, so say a human brain, and you isolate and disassociate every cell. And then every cell results in one observation, and that one observation has up to 20,000 features. After you do the sequencing, we do this alignment step where we essentially map the sequencing reads or from the FASTQ files to some kind of reference genome or transcriptome. So here there are 18 different reads mapping to the reference gene 1 for cell 1. There are 18 little black lines, please trust me. And then you record that in a matrix. So that matrix simply contains all the genes that were being expressed at the cell at the time that we sequenced it. What you know about this matrix compared to what's known as bulk RNA sequencing is that that matrix is very, very sparse. So in bulk RNA sequencing, you just get an average of expression across all the cells in the tissue. But for single cell, that matrix can contain anywhere between 50 and 99% zeros. It is quite sparse compared to bulk RNA-seq, where it's maybe less than 30%, not so many zeros. Nonetheless, you can do some pretty amazing things. You can plot your data. I'm going to show you a lot of plots today. You can project your data into a low-dimensional space, such as along principal components, and see how the cells separate, corresponding to different cell types. So next, I'm going to go into give you an overview of some big computational challenges in the field of single cell and give you a little bit of a flavor of some of the solutions that we have been working towards. I'll take a deep dive on one of them, but most of the rest, I'm just going to give you kind of an overview. So in terms of single cell RNA sequencing, why is this so hard? What is the big deal compared to bulk RNA-seq? Well, there's two aspects. One is simply generating the data. I'm not going to touch on that. It is a lot harder to generate single cell RNA-seq data. It is more complicated, but you get better resolution, meaning you get to see these different types of cells. I'm going to focus on the computational analysis, or the computational challenges for analyzing these data. So the first challenge is that when you move from bulk RNA sequencing to single cell RNA sequencing, you just simply have a lot more variation that was hidden whenever you're averaging across lots and lots of cells. Suddenly, when we go down to the actual cell level, the cells are doing crazy things. They might be going through the cell cycle. They might be being told to go through programming or cell death. There are technical reasons that introduce a lot of biological variation, often due to the kind of experiment that was used to generate the data, or there's more biological variation. So on the technical side, for example, the RNA is always going through a degradation process. And so you might have some RNA in your cell that is unfortunately going through the degradation process. And if that's happening, you will not get a lot of RNA in your cells. There's another technique or another technical bias called amplification bias or PCR bias. Essentially, the amount of RNA that we get in a cell, one cell, compared to a bulk RNA-seq sample is very, very small. So we have to amplify it up. We have to make tons of copies of it. But let's say I have two genes, one that's very lowly expressed and one that's very highly expressed. Put both of them in like an urn, like think red and blue balls. If I have 90% blue balls and like 10% red balls, if I stick my hand in the urn, is it likely that I will get more blue balls or red balls, for example? Blue, right. Now let's say it's 99 blue and 1 red. Is it likely that I would get the red? Yeah. So what we do is we just amplify everything up. We make tons and tons of copies of everything. But that introduces particular biases that unfortunately make it challenging for single cell. Then there are things called batch effects. So batch effects are essentially me and Ting go into the lab and I generate some single cell RNA-seq data on Monday. Ting generates some single cell RNA-seq data on Tuesday. On Monday, I have the sniffles. On Tuesday, Ting does not. Or the weather is rainy on Monday and the weather is not rainy on Tuesday. There are 1,000 ways that things that happen to the lab suddenly show up as changes in gene expression that are unwanted to what we actually care about. But it's actually a really big problem. So in 2015, I wrote a paper talking about best practices for experimental design. Believe it or not, when we first started to generate these data, people were generating very confounded experimental designs, and it made it challenging to try to tease out the biological versus technical signal. So moving to the biological variation, there are a whole other set of issues that introduce variation that's masked when we do the bulk RNA-seq. So for example, different types of RNA will degrade at different rates. Environmental stimuli, maybe you sequence some cells and they were currently responding to some environmental stimulus in the tissue. Or maybe they were going through some kind of temporal process. We just take our hands and we extract out the single cells at that one point in time. We don't actually get to see the full temporal resolution. Or maybe we extracted out cells from one region of the tissue versus another region of the tissue, and there's spatial variation there. There are 1,000 reasons why biological variation causes problems for single cell. Yes?
Just a question about the amplification bias.
Yes.
The way you describe it first to me is just variability, but it's not bias. It's just amplified by the same portion.
Yes. So until about 2018, that was correct, but we would get essentially a lot more reads for the highly expressed stuff. And we wouldn't actually get a true count of mRNA. We were just getting reads that were getting amplified. So if you did 16 rounds of PCR versus 17 rounds of PCR, you would see differences in the expression. And you don't want to find differences simply due to how many rounds you do, for example.
So it's not kind of proportional. Still, the higher ones bias even higher.
The higher ones are typically biased, and the lower ones are very hard to sequence. Yeah, in general. Good question. So the second big challenge is around simply the volume of data. So to try to put this in perspective, when we first started in single cell RNA sequencing, well, there was one cell. We did our first single cell RNA-seq sampling in 2009. We sequenced one cell. Yay, us. Then in 2014, 2015, commercial platforms became available. So this started to become widely implemented in lots of computational labs. But we were standardly generating a data set of 96 cells. So 96 is more than one, but it wasn't, thousands. Fast forward to 2015, 2016, we were generating thousands of cells per data set. I don't have a plot. Unfortunately, I should make a plot. But now we are standardly generating data sets with millions of cells. So if you think about that, if I have a count, like a table, a matrix of counts with 1 million observations, or N is 1 million, with 20,000 features, that's a pretty big table. Depending on what kind of technique you're using, computational method you're using, you're gonna have to load the data in memory. You might have to do some additional storing of maybe distances between the cells, depending on what you're doing. So as the data become bigger and bigger, you need more memory, more storage. It takes longer to run things. It makes things more complicated. So if you go to try to extract out data from the CCI cell by gene API, they actually have 85 million cells for you to play with across 1,300 data sets. And they claim to have sequenced and identified 844 cell types, which is cool. So let's say, for example, I want to go pick out liver cells from this data set. I'm going to guess there's 10 million cells that are liver cells in this data set. And then I want to identify cell types, subsets of cells for different types of liver cells. So I want to perform unsupervised clustering, such as k-means, and I want to identify what are the different kinds of cell types in my liver cells. So I hope everybody's familiar with k-means, but I'm just going to briefly go over it in case you're not. So if I want to do k-means, which is a widely used unsupervised clustering algorithm to take a data set X with n observations and partition it into k groups, that's the goal of k-means. If I want to do that, formally what k-means does is it tries to minimize this within cluster sum of squares criterion, where X is our matrix, and we're interested in estimating the parameters mu I. They're often called centroids, or the centers for each of the k groups. And we want to go about estimating the parameters. So this turns out to be, in practice, we don't actually go about trying to minimize this criterion. We use just a simple expectation maximization algorithm. It's called Lloyd's algorithm. It's the base algorithm in Python and R. It's an iterative algorithm. So the idea is the following. Let's just randomly assign centroids. Like I'm just going to randomly simulate the centroid values, mu I. Or you can do this more cleverly, but let's just assume it's random. You take each observation and you assign it to its closest centroid in a high dimensional space. That's step one. And then step two is you take all the observations that have been assigned to group one and you compute the new centroid. And you do this for observations in 2 and 3 and so forth. This works really well in practice if you have a small data set. If you have a large data set with 1 million observations, one, you have to load all the data in memory. Two, you have to load all the data. You have to calculate all the pairwise distances between each observation and its closest centroid. That's 1 million by 1 million. That's, imagine going to 10 million now. So we're thinking about scaling up the task of performing unsupervised analyses. So instead of an EM algorithm, you can actually use stochastic gradient descent for estimating these parameters, mu I. And the algorithm is called mini batch k-means. You can actually use mini batches in many areas that use stochastic gradient descent, but here I'm using it for k-means. And the idea is the following. So if I have 10 million observations or 10 million cells, let's say I have all the data stored on disk. I don't just go to Python or R and load the data naively. Let's say I load in 1% of the data into memory. They call that a mini batch. It's a random subset of the data. And then I estimate my centroids. I spit that out of memory. And then I load in the next 1% of the data. I update my centroids and I spit that out of memory. So I traverse over the whole data set to estimate my centroids. And at the very end, there's like one more assignment step where I assign each observation to its closest centroid. So the nice thing about this is like you don't have to store the data in memory. You only need to store the 1% or 5% of the data. This is a user-defined parameter. And then only the distances between the observations in the mini batch and the K centroids. And this is like a natural candidate for really scalable clustering. So I won't show you all the results. I'll just show you this one that really blew me away the first time I saw it. So the take home is mini batch k-means is ridiculously fast and memory efficient. So we took a data set with 1.3 mouse neurons and we downsampled to smaller sizes of data set, starting on the x-axis from 75,000, 100,000, 300,000, 500, up to 1 million. So these are just smaller sizes of the same data set. And on the y-axis, we tracked two things: the max memory used over the course of the algorithm, and then the elapsed time it took. We compared three things. One, just vanilla k-means with Lloyd's algorithm. And you can see that it's pretty memory intensive as you get larger and larger data, and it's actually kind of slow. So that's unfortunate. But if you don't want to deal with data on disk, maybe you can say, Stephanie, I have a big cluster, I can load my data into memory. Even if you want to do that, that's the green line. So that's mini batch k-means loading in the data into memory, but you're computing all those pairwise distances with only 1% or 5% of the data at a time. So you get a huge cost savings in terms of memory and speed as the data become bigger. And then in blue, if you keep the data stored on disk the entire time and you just load in 1% of the data at a time, you can cluster 1 million cells in under 2 gigabytes of memory in 10 minutes. And I'll just say that this line will continue to stay pretty much flat as you go along because you're thresholding the amount of data that you load into memory. If I'm only loading in 100 cells at a time, that will never grow. It'll just continue to be flat, which is really cool. So that is how we started to handle large volumes of data for unsupervised clustering. Next, I want to talk about a different challenge around dimensionality reduction. So a really big challenge in single cell RNA sequencing is the fact that when we sequence a cell, we don't actually sequence all of the RNA. It's surprising, but we only get around 10 to 50% of the RNA. We're not super good at extracting all the RNA out of the cell. And so what happened was, whenever you only sequence, say, 10% of the RNA, the field thought and said, okay, so we have zeros, maybe that represent the gene was being expressed at the cell at the time that it was sequenced, but we just didn't stick our hand in the urn deep enough, or we didn't sequence deep enough. So we get a zero that should actually not be a zero. Or maybe the gene was truly not being expressed at the time. So there are these different kinds of zeros, but in general, single cell RNA-seq is super, super sparse. And this had a huge debate in the field at the time. It was like for 5 years, people thought, well, let's treat it like bulk RNA-seq. Let's log transform and normalize our data, make it look Gaussian, and then we can be off and running with everything that assumes a Gaussian model. And some people said, well, these are counts, right? And we have a lot of zeros, so maybe we need zero inflated models, a zero inflated Poisson, a zero inflated negative binomial, zero inflated Gaussian, like you name it, a lot of zero inflated models. And then it turns out it actually depends, the model that you choose to model the data actually depends on the kind of single cell RNA-seq data generated, believe it or not. So there's not one model to rule them all, unfortunately. Depending on if you sequence, for example, droplet-based data versus full-length data, different models are actually designed to be better. But I can tell you that sparsity, no matter what, impacts downstream analysis. So I want to talk about PCA because I've now picked on PCA a few times. I've talked about, let's take our high dimensional data and just project it in a low dimensional space. This is no problem, right? What's wrong with this? So I'd like to tell you what's wrong with this. And specifically, I'd like to do this in the context of dimensionality reduction. So bulk RNA-seq, in bulk RNA-seq and in microarrays, the previous technology before that, we apply a set of normalizations and transformations to the data to kind of make it look Gaussian. We do that for a variety of reasons. So I'd like to tell you why this is problematic in single cell RNA-seq, and then I'd like to introduce a technique that we developed to correct for this. So this is a distribution of expression for one gene across a set of cells that we don't expect much biological variation. They're like very similar, not a lot of biological variation going on. Does anybody tell me what that distribution roughly looks like? Does it look Gaussian? Anything? Exponential? Okay. It's integers, I can tell you. It's 0, 1, 2. What? Poisson? That's a good one. Anybody else? Maybe a little negative binomial? A little over dispersion? Okay, but you get it. Counts, right? These are counts. So what people do is for every cell, the strategy is normalization. We want to normalize for how much total RNA there is in a cell. So we just take all the counts for a given cell and we divide that count by the total amount of RNA. And then we multiply it by 1,000,000. It's a long story why we multiply by 1,000,000. It's absurd to do this, but that's what it is. So we have this plot in the middle. It's called counts per million. It's the same as this, except I have applied the transformation counts per million. Again, I just divided every cell by its total amount and multiplied by 1,000,000. So does anybody want to say or see what the difference is, or try to hypothesize what the difference is between those two distributions? Are the zeros still 0? Yeah, and everything else, what happened to it? Looked like it moved over to this direction, the right? Yeah, okay. So then the last step is called a log transformation. It has a lot of historical reason why we do this log transformation. It's a form of variance stabilization if you want to know. But whenever we do a log transformation, we take the counts per million value. We do a pseudo count of 1. Again, this is like a lot of history here, why we do the pseudo count of 1. And we just apply a log2 transformation. So what happens here? Zeros remain 0, right? Zeros are still 0. And then here, everybody squint. Do you like this? And then would you agree that that's Gaussian-like? You got to squint really hard. Okay, so this is what the field has gotten to. They said, look, we have something that's Gaussian-like. We're ready to do PCA, right? So this is what has happened in the field of microarrays and bulk RNA-seq for decades. But the difference is this. Single-cell RNA-seq data is sparse. It has 50 to 99% zeros. Bulk RNA-seq does not have this. So does anybody want to take a guess what will happen when you apply PCA? Not a lot of guessers, huh? Okay. So if you apply PCA to this data, again, we expect not a lot of biological variation. These are monocytes. They should be a big blob, a big random blob here. If I'm a biologist, let's say, Ting, you're a biologist. Have you discovered something new? Are these cells suddenly different from these cells? Does it look like a blob or does it look like it has structure? It's like structure. Yes. So a biologist would say, I've discovered 2 new cell types in my liver data set. This is amazing. But in fact, what you have discovered is every cell here is colored by the fraction of zeros that are in that cell, essentially the number of detected genes. That's the difference between single cell and bulk RNA-seq. This is hidden in bulk RNA-seq because again, it's a mixture of cells, but in single cell, it's like beautifully shines. So the challenge for applying dimensional reduction techniques that were never designed for single cell data are that you get these false positive kind of results. And I can tell you, this is still the standard in the field today. So we thought about this and we said, okay, so you guys threw out some great distributions earlier, Poisson, right? Let's say we want to try to model these data and see if Poisson is actually a good model for them. So what we did was for every gene here, every dot here is a gene, we calculated the empirical mean on the x-axis and then just the empirical fraction of zeros along the y-axis. And then we fit models to it. We actually fit 3 models, a multinomial, Poisson, and a negative binomial. Does anybody want to take a guess if they think that's a good fit? Yes or no? Do you think, is a Poisson model a good fit here? Does it follow kind of what we see in the data? Yeah? Yes. That answer is yes. It's definitely yes. We saw this and we were like, amazing, great. We have models that count-based models that we can use to model these data. So we got inspired by this and we said, okay, can we generalize the concept of PCA for non-negative integers or non-Gaussian data? So if you think about the PCA objective function, principal components analysis, the objective function formally is trying to minimize this mean squared error between Zij, which are the normalized log transformed data that I talked about before for the ith cell and the jth gene. And we want to minimize this criterion to between the data in some kind of low rank representation, Ui, which represents typically some kind of factor for the ith cell, and nu-j, which represents some kind of loading for the jth gene. So we're interested in trying to estimate Ui and nu-j. So if you stare at this objective function, you can notice that the minimization that I just talked about before of the mean squared error is equivalent to maximizing the likelihood of a Gaussian model. So to be clear, PCA doesn't require any kind of formal Gaussian assumption. It's just that you stare at these, they're similar in the sense that those two things are equivalent. So we said, can we essentially replace the Gaussian model with, for example, a Poisson model and model those counts directly? And that is what turned into something called GLM-PCA. It's available as a package if you want to try it out. It assumes different kinds of models on the outside, and the goal is to try to estimate the low dimensional representation, Ui and nu-j. In practice, we actually go about estimating these parameters a slightly different way, but if you're interested in kind of like the details, I definitely encourage you to check out the paper. So just to bring us back to like this negative control data set where we don't expect a lot of biological variation, remind ourselves, we found this like beautiful structure where we don't expect any kind of biological structure when we apply PCA. In contrast, when you use GLM-PCA that models the counts directly, that's designed for single cell RNA-seq data, you get a blob, which is beautiful. I was very happy to see that. Yes.
It looks like the first and second direction capture pretty much the same amount of variability. Do I still have some properties like some sort of order, natural order in terms of how these species are ordered? And
The cells?
The PCs in terms of explained variability, and in what sense they are good approximations to the original data.
Oh, the GLM PCs?
Yeah.
Oh, can I talk to you about that afterwards?
Yeah.
Okay, that's going to be a little bit to explain. Okay, and then the last challenge I wanted to talk about for single cell RNA sequencing is just simply the lack of documentation and resources to do analysis for this data. So when I started in this field, it was very fast moving. Not a lot of people were generating resources to help people analyze the data. So I spent some time actually with Rafael Gottardo, who unfortunately is not at the front bench anymore. But we worked together to essentially write a book. And so it turned into this paper thing at first. And then we talked about ways to standardize ways to, and we built standardized ways to store single cell data and then workflows to analyze single cell RNA-seq data end to end, starting from like experimental design, pre-processing, quantification, normalization, dimensionality reduction, clustering, report creating. We really tried to be very thorough. But the thing I'm most proud of is this online book. So it's written in Bookdown, if you are familiar with it. It's built very periodically on the bioconductor build system. It has a lot of cool things I like about it. One of the things I really like about it is it has good philosophical discussions. So for example, one question I get from my collaborators are, what are the true numbers of cell types in my data? Tell me the true number of cell types. And the answer is there's not a true number of cell types. If you apply k-means with a k of 5, k-means will partition your observations into k or 5 groups. You apply k-means with a k of 6, it will partition your data into 6 groups. It is not designed to tell you what is the true number of cells. It just is designed to partition your data. And then there's lots and lots of code. So this has been really helpful to me and my collaborators. So I share this with you in case it's useful for you to analyze single cell RNA-seq data. Okay, any questions before I move into the next part of the talk? Yes.
For the issue with the PCA,
Yeah.
does it matter how many highly expressed genes was select? For example, if we choose a smaller number of highly expressed genes, hopefully it would be less zero information.
Well, yes. So the question is, does the choice of feature selection impact downstream analysis? The short answer is yes, but also which genes you pick generally impacts your downstream analysis. So if I don't have informative features as input to PCA, PCA will never be able to help me parse out what the cell types are in my data set. So you tend to lean on the side of wanting to include thousands of them. And when you get to that point, you get a lot of zeros no matter what. It's a good question. Okay, is it okay if I turn off the room lights for just 2 minutes? Yes.
So you mentioned for every cell, only 50% of the RNAs are read out.
10 to 15%
10
is,
to 15.
yeah.
So is that just random or just designed?
That's a good question. I mean, part of it is cost. So essentially, when you do single cell RNA sequencing, you pay for the number of sequenced reads. So if I have lots of money to spend, I will just keep sequencing and sequencing and sequencing, and I'll get the highly expressed stuff first, and I'll get the lowly expressed stuff later on. But if I don't have a lot of money to spend, I'm gonna get less than, I'm only gonna get the highly expressed stuff. Does that make sense?
So that's why it's device.
Yeah. So I just want to does anybody have a concern for just 2 minutes? Okay. Is that okay? Okay. So I was going along in my job thinking everything was fine. Single cell was my career. And then in 2020, this new type of technology dropped on my doorstep, and I wholeheartedly switched my entire lab to starting to develop computational methods for this data. So I want to spend the rest of my time today talking to you about the type of data that's out there and why I really feel like it's a really great opportunity for statisticians to get involved, bridging these fields of spatial statistics with high-dimensional data. So this is an image of a postmortem human brain tissue, and it's showing you what's called the hippocampus. So this is a particular brain region that's highly influential in many different fields. It's related to memory and learning, also very important in Alzheimer's and beyond. So the image is the following. This is what's called the granular cell layer, shown in kind of the curly part. It's a very dense set of cells that have been lit up and tagged with a particular type of protein. And then here in the center, this is called the CA4 region. This is called the CA3 region. This is called the CA2 region. There are domains for the rest of the tissue, but I just wanted to generally tell you that this is a very well-studied region. And the morphology of what you see, the granular cell layer, the CA342, is very well structured. It's the same for me, it's the same for you, it's the same for everybody in this room. And so because it's highly implicated in so many disorders, we are interested in sequencing it. Now, this, for example, right here is one neuron. And what I can get from this data is at this one neuron, I can capture genes that are expressed in this particular neuron, and in this particular neuron, in this particular neuron. In this type of technology, you only get a couple hundred genes, but in other technologies that I'll talk about next, you can actually measure the full transcriptome. But the difference is their level of resolution. So in this particular data, I actually get subcellular resolution. I actually get counts of where the RNA was in an XY plane, and I can aggregate it together, and I can count up all the RNA in a given cell, for example. Now, it's not just me who's really excited about this. So this particular type of technology called spatially resolved transcriptomics was labeled by Nature Methods in 2020 as the method of the year. And there have just been tons of opportunities since then, and I want to tell you more about it today. Okay, so now I'll put it back on presentation lights. There will be one other part of the presentation where I'll ask to do that, but for the most part, I just wanted to like showcase the data in its entirety first. So on some level, you can think about spatially resolved transcriptomics as kind of an extension to single cell RNA sequencing. So the idea here is, bear with me, I have kids, there's a lot of Legos in my life. If you think about bulk RNA sequencing, imagine it's like a big pile of Legos where you measure, like say the width of every Lego. That's a feature that you have for that big pile of Legos. If you move to single cell RNA sequencing, you get information about like the cell types, the proportion of cells, things like that. Spatially resolved transcriptomics is kind of like being able to put together the RNA in a 2D space. There are different kinds of technologies, two really broad categories. The first one is the imaging technology. That was an example that I just showed you a moment ago. It's from the 10x Genomics Xenium platform. And then the one I'll talk a lot today about is capture plus non-targeted sequencing. So we get to capture the full transcriptome within these little, what are called spots. So you just lay the tissue on the slide, all the RNA binds to the slide, and then you just wash off the slide, all the RNA from the slide, and you sequence it. So because I think it's important to understand the data, I wanted to just go over briefly the platform. So this is called a Visium capture array, and it has 4 different capture areas, meaning that, let's say I have a postmortem human brain, and I have a 3D tissue block. If I do one deli slice, I can put that deli slice right here. And I can take a second deli slice and put it here, or maybe from like a different brain or a different cohort, but you just lay the tissue there in the 2D space. If you zoom into one of the capture areas, it's outlined by what's called a fiducial frame. The fiducial frame is important for aligning the gene expression with an image. The image could be an immunofluorescence image. The image could be a bright field image, such as a histology or an H&E image. But you now are getting multiple modalities of the data that you can use for your downstream analyses. Within the fiducial frame, you have what are called spots. These are these little round things that I showed you before. You have up to 5,000 spots within a capture area. And the whole fiducial frame here is tiny. It's like 6.5 millimeters by 6.5 millimeters. So it's not super big. And then within a given spot, you have what are called primers. They're little barcodes that are sticking up on the within a given spot. It's essentially a locator for what is the XY coordinate plane. So all the RNA that binds to, for example, this spot is going to have a spatial barcode. So I can map back where the RNA came from when I sequence it off. Does that make sense? Okay. So thinking about data structure. So if I think about single cell RNA sequencing or single nucleus RNA sequencing, typically what I have is for a given set of cells, I have a set of counts incorporating, for example, the amount of RNA expressed in each of the genes. If I now move to spatial transcriptomics, you still have that same kind of matrix, except instead of a cell, it's at the spot level. You can have multiple cells within a spot. You also have XY coordinates. This is what the spatial barcode looks like. It's just a series of A's, T's, C's, and G's. And then you also have images. So for anybody interested in image analysis, you can, for example, see the hippocampus right here in this mouse half hemisphere. It's so well studied. And it's so lively. This is like a very dense set of neurons, for example, that I was showing you in the GCL layer. So the data is more complicated than single cell in the sense that you have all this other information that you have to deal with. So I want to talk to you a little bit about one particular project that I originally got brought in on to analyze the data. So we took 3 postmortem human brains and we took a tissue slab from the anterior region and we just laid that slab down in a 2D space. If you look at this black box, this is the box of tissue that we actually cut out from the slab and the thing that we put on the capture area to sequence. So it's a very, very tiny amount. Morphologically, this region of the brain in the dorsolateral prefrontal cortex, very well studied. Essentially, it has something called white matter and then 6 layers of gray matter. I will give you examples of that in just a moment. So if I took one postmortem human brain, the experiment was the following. We did 1 deli slice, we laid it here. We did a second deli slice, we laid it here. And then we moved into the tissue block 300 microns, and then it did another 2 tissue blocks. And then we repeated this for 3 different brains to get a sense of the variation that we would see across spatial replicates and then across donors. So if we want to visualize it, this is what the H&E image or the histology image looks like. The darker area in the histology image is actually what's called white matter. And the lighter image, the lighter pixels are what are called gray matter. So the gray matter contain all the neurons, for example. And I can plot then gene expression. So SNAP25 is a marker for neurons, and you can see it lights up where the layers 1 through 6 are. MOBP is a marker for white matter. It lights up where the white matter is. This is a really cool gene called PCP4, and it's a marker for synaptic plasticity. But you can see it like lights up in a very non-random way. It's not like it's totally random across the slide. So if you want to see the kind of subject to subject or donor to donor variation, that's what I'm showing you here. So I'm just showing you the gene expression for one gene, PCP4. So this was like deli slice 1, deli slice 2, moving into the tissue block 3 and 4. But you see that you get things that are not going to help you line up the tissues in a z-axis very easily. Again, the tissues are wet, they're tiny, they're being put on the Visian capture area like by graduate students who are very nervous. These are very costly experiments, you know, so like a lot of things can happen. It's not perfectly the same, but you might be interested in saying, well, maybe I want to line these up in a z-coordinate plane so I can start to do statistical inference across donors, for example, in a neurotypical control group versus an Alzheimer's group. So this registration or alignment is a really big open challenge in spatial transcriptomics right now. So I promised you the layers. So this is that same tissue section that I talked about with the white matter shown in black and the layers 1 through 6. I got this annotation from a very kind scientist who manually annotated, to the best of her knowledge, the layers of what we expect for these images. So you can kind of think about it as a gold standard, right? So what we did was we said, okay, so let's apply an unsupervised clustering algorithm like k-means. How bad is it? Or how good is it at guessing what these layers are? So you want to take a guess? Good, bad? You think pretty good? You feel confident about k-means? No? Okay. So this is graph-based clustering. And we also tried k-means, did not work so well, unfortunately. Semi-supervised was kind of a supervised learning algorithm that we learned. We asked what are the differentially expressed genes, we built a model, and then we tried to predict. It didn't work so well. So this was all in 2020 that we were generating this data for the first time. And at this time, I felt very passionate about trying to make this data as easily accessible to the research community because I really wanted methods developers to get involved. I wanted them to leverage this data and get super excited about spatial transcriptomics. So we made a package, we wrapped it up in an R package, and you can download the data with 1 line of code. It was amazing. And remember, this data has manual annotations. It's well annotated data set. Maybe you want to develop a new spatial clustering algorithm and you want to evaluate, you know, the domains that you are predicting compared to what the gold standard says.
Can I ask, what are the annotations based on some like reading the histology or
Histology and marker genes.
Okay.
Yes. It should not be labeled a gold standard, but I'm using that kind of informally.
Okay.
So that's exactly what happened. The first spatial transcriptomics or spatially aware clustering algorithm came out. It's called BIOST. They took the annotations directly from the data set, which we knew was gonna happen. And then they did the thing that we were expecting for them to do, where, they compare event clustering, model-based clustering, these other algorithms, and this was their predictions. This is very sketch in my mind. I've never been able to make Bayes look that good, but it's another story. So they compared, their clusters to the manual annotation. They got a paper in Nature Biotech. It was amazing. Anybody want to guess what's happened next?
Another map.
So this is Bayes, right? Nature Biotech, that one tissue sample appears, right? So in case you're curious, lots of people have started to develop spatial clustering algorithms. So there was a method from Mingyao Li called SPA-GCN that was developed. Anybody recognize that tissue? Then there was another one called precast. Actually, precast is really good. I strongly recommend using it if anybody's interested in knowing more about it. This is what we're using in a lot of our data set analyses projects right now. Then there was one called setter. Do you recognize that tissue? It's like in my nightmares. Deep ST and graph ST. True story. There were actually two deep ST algorithms that were pre-printed right around the same time. And it took me like weeks to figure that out, that they were not the same algorithm. My student kept coming to me like describing one of them. And I kept thinking, I was like, what are you talking about? It's not how it works. And then we figured out there were actually two deep ST methods that were pre-printed. And I just tweeted it out. I was like, FYI, anybody interested in knowing this, there's actually two different deep ST methods. And then like 6 months later, the authors of Graph ST, who I do not know and I've never met, replied back to my tweet and was like, we changed our name. And I was like, whoa. Also, thank you. Like, thank you for the field. So I just want to make the point that, I continue to see this tissue appear everywhere in my life. This is not an inclusive list. And I think the reason that this happened was the following. So if you really want to motivate computational tool developers, in my mind, one of the best things you can do is to manually, to curate a data set and to wrap it up in a package and make it as easily accessible to the community so that they don't have barriers for downloading the data. And it really builds trust. And yeah, so strong recommendation for doing this, but I don't know if I would do that exact same thing again. So part of one of the tasks in that study that we were interested, that we were tasked with was, okay, so you have these tissues. We know PCP4 is important. You can see PCP4 is like beautifully spatially variable in a non-random way. Can you find the spatially variable genes in a data-driven way? The ones that we don't already know about. So the term spatially variable gene was new to me. It took me a while to try to understand it. Unfortunately, we still don't have a good definition of it as a community. There was a paper called Spatial DE that was published in 2018. They defined spatially variable genes as genes with any kind of spatial pattern, essentially. And if you read about what they mean, they essentially mean that the spatial pattern is being quantified under a specific model. In their particular model, they are using a Gaussian process regression model, which is a widely used regression model in geospatial statistics. So we did that, and we found a set of spatially variable genes. These ones we already knew. They're associated with the layers. Does anybody want to guess what these are? Do they look spatially variable to you? Does it look like noise? No guesses? Super quiet. They are associated with immune and blood. So for example, you can imagine a blood vessel right here, and you can see the genes that are being expressed for that blood vessel. So you have different kinds of spatially variable genes. So if you stare at, these plots enough, you can see sometimes the genes are associated with the morphology, and sometimes they're more associated with like little niche things that are happening in the tissue. So there were a couple of problems. One, we quickly noticed that all the tools that were developed at the time, they basically assumed that this kind of variation or bandwidth was going to be the same for every gene. That is clearly not true. And then the other problem was this tool was painfully slow. So spatial DE fits a Gaussian process regression model and is actually cubic as a function of the number of spatial coordinates. So we have 5,000 spatial coordinates here. And you could imagine as if we ever go to 1 million, which by the way, we just did last month, this is going to be insane. You cannot use a standard Gaussian process regression model. For every gene, it took us over 30 minutes to fit these models, and we had thousands of genes that we did this for. So that motivated us to develop a fast version of this. And the goal was to be linear as a function of the number of spatial coordinates as opposed to cubic. So there are a couple approaches that you can use to make Gaussian process regressions fast. One is to assume some kind of low rank structure, approximate the data at a lower number of spatial domains. That has its pros and cons. And then we actually ended up going with something called nearest neighbor Gaussian process. So the idea was it was introduced by Adidata in 2006. You just use an approximate likelihood using a set of nearest neighbors, like roughly 10 to 15. And it approximates the full data decently well, and it gives a sparse precision matrix. And it's also, the most important part is that it's linear as a function of the number of spatial coordinates as opposed to cubic. So let's say, for example, you plot this gene, PCP4. We know that it's spatially variable, but just bear with me. The idea is the following. You have normalized log transformed data at each of the spots for a given gene, and we're going to fit these models per gene. So the model that we fit is a nearest neighbor Gaussian process. So it has a covariance matrix, that is approximate covariance matrix, parameterized by a parameter sigma and tau squared. So this is the approximate version of the covariance matrix that's actually approximating the full covariance matrix with some kind of covariance function, parameterized by the parameter theta, and then also tau squared. So tau squared in the context of, if you're familiar with spatial statistics, is often referred to as the nugget. I still don't get the history with that, but it's often referred to as the nugget or the non-spatial variance. And then the spatial variance is parameterized by sigma squared, where this covariance matrix, or C of theta, is often referred to as a kernel, for example. This is an exponential covariance matrix. You can have different kinds of covariance matrices. We're interested in estimating 3 parameters: tau squared, sigma squared, and L. L is a very particular parameter that most people just usually fix. We said, well, this seems pretty important. Like, L is a gene-specific parameter that allows the bandwidth for different kinds of variants to appear in the data. In terms of assessing statistical significance, we do a very vanilla thing where we take the likelihood ratio statistic, comparing a linear model with a spatial component against a model without a spatial component to rank the genes. And then we just approximate that likelihood ratio test using a chi-square distribution. And then we also calculate an effect size, which is the proportion of spatial variance divided by the spatial variance plus the non-spatial variance, or the nugget. And so genes that have a high proportion here are meaningfully interpreted as having more variation than what you would expect under just random noise. So if you go back to these 6 genes that I showed you before, the ones that are associated with the layers versus the immune and blood, the estimated length scale, so this is a distribution of all the estimated L's for each of the models that we fit. PCP4, SNAP25 at Omobuki, they all have a large L that was estimated. And then the 3 genes over there, they all have a small L that was estimated. And this is one of the things that really gave our method an edge, in the sense that other people basically just fit this parameter and they don't worry about it, and they just estimate tau squared and sigma squared. But it was actually really important in terms of actually getting meaningful genes. How does it perform? So there was a benchmark that was actually carried out in December of 2023. Look at all these methods that have come out for spatially variable genes. It's a lot, huh? So NNSVG is shown here in the lime green. Really wish they'd picked a different color. So it's memory efficient, it's fast. We knew that, and it was really nice to see independently verified. The authors also went and tried to evaluate a form of accuracy. It performs really well, so that was really exciting to see. If you want to see the benchmark, the simulation studies that we did, you can check it out, but I just thought of showing this in the context of the other ones made a lot of sense. Okay, so am I out of time? Yes?
Yeah.
Okay, 2 minutes? All right. So I want to show you something, cool, let's see. Okay, so the last part I want to talk about is these images. So this is spatial transcriptomics data. It's super, super cool. If I want to, for example, have a collaborator explore these images, they have to do the following. They have to use what are called GUIs, or graphical user interfaces. The two big ones are ImageJ and Fiji or Napari. They are software that you have to download locally. You load in the images. You can only load in like 1 or 2 at a time. They crash if you try to load in more. There are many reasons why this is very challenging. Wouldn't it be nice to just hand your collaborator a URL, like a link, and to be able to interactively explore these data? Similar to the idea of Google Maps. So whenever you are interested in using Google Maps, you hit the app, you are loaded into a map of where Seattle is, you start dragging it, you get more data downloaded from an AWS bucket of the city next door. We wanted to do the exact same thing for spatial transcriptomics data. So we built this thing called Smooey Browser. I will not go into the details, and I will just show it to you because I think it's cool. Oh, I promise you room lights off. Is that okay? Okay. So let's see. So if you go to Smooey Browser, you can do this on your phone too. It's so cool. The idea is we just have some dummy data here that you can load in. This is all live. This is like UW internet. This is not me showing a recording. So you can, for example, let's turn off the genes. Now I'm zooming in to, downtown in Seattle. And what I'm showing here is, oh, this is harder to do than I thought. Okay, so the blue represents the nucleus of a given cell. So these are all cells in a particular tissue. I'm now going to turn on a protein that tags the neurons. Can you guys see which ones are the neurons? So this is what's called an immunofluorescence image tagging different channels. So this is microglia. If you try to segment microglia, it's like the bane of my existence. It is not easy to segment. Oligodendrocytes are shown in light blue. GFAP or astrocytes. So what you get is a set of proteins in a 2D space. And you can see, for example, neurons light up in one area. Oh, let's see. Neurons light up in one area, while astrocytes light up in another area. Do you guys see that? That's information that you can actually use in your clustering algorithm that people are currently leaving on the floor. You can also do the following. So because this is Visium, you can turn on all the spots. This is the 5,000 spots for the given tissue. You can actually directly see what cells or how many cells there were in a given spot, and you can know what they were, for example. And then if you want, you can go and search for your favorite genes. So you can do, it's really hard to do with my head turned. PCP. Oh, that's not PCP4. Hold on. PCP4. Nope, not R. Nope, I really can't do this. I'm not a perfect. Okay. All right. That's PCP4. That's layer 5. You can turn on MOBP. Yes, that one. And that's white matter, for example. So you can actually go and interactively have your collaborators explore this. This is all open source. It's based in Python. You just throw your data either can be locally, and then you interactively explore it in your browser locally, or you could throw it in an AWS bucket, give it a URL, and it will interactively download data as you drag it along. So this is really cool. But it's a very similar idea to Google Maps, but just for spatial transcriptomics. Okay, and then I was told I can, for example, go. Okay, that's it. Thank you, everyone.
You know.
Yes.
I mean, do you have a sense of like how that be variable or not impact the genes that would constitute?
How, could you say it again?
How setting the values of like scale theory impacted the genes that would constitute spatial variable versus not?
Yeah, so we did that. Basically, you get nonsensical genes. You get things, so things that are kind of like in the middle or near the mode, that's what it typically finds, but it misses the things on the edges. And also like in terms of how it's ranked, like it's essentially a ranked set of genes that you get. The rankings also, like you would expect certain types of genes to be at the top and we weren't finding those genes at the top. So then we said, hey, let's discover it with the data, and that fixed the problem. Thanks for the question.
I have a question. So you have in the Gaussian model, you also have X times beta.
Yes.
So you can use regressors like covariates. So for example, like I think some of the spatial variation might be due to like cell types or like that. So how do you use those?
So in terms of the X beta in the mean component of the nearest neighbor Gaussian process, we are thinking about it. And well, one way we definitely use it is to adjust for the overall mean. So certain genes are just more highly expressed versus lowly expressed. That is uninteresting variation. So we can just directly adjust for the overall mean, for example, in a covariance matrix. So any kind of like fixed effect, you can just stick there. Or for example, you maybe want to find, like you define a layer, like you define layer 5 and you want to find variation within layer 5. Like you want to find high expression here and low expression here. You can use the covariate or the design matrix to specify like the location that you're interested in finding the variation.
And do you consider X being like the cell type in that location? Because that could explain why these genes express.
Yes, so that's a great question. So I think what you're getting at is a spatially variable gene could just be differences in cell types. And cell types have clear differences across the 2D space. So depending on the kind of spatially variable gene you'd like, you will find just differences in cell types, or in some cases, you'll find variation beyond just differences in cell types. This is one of the problems with the definition of spatially variable genes. It's an ill-defined problem at the time, or at the moment, so. Okay, thanks everyone.
No more questions. Thank you, Stephanie.
Sure.